In this video, I want to go through the biomolecules that you are going to be required to know in Lab 2, and we're going to be using the actual models that you're going to be seeing in class. There will be other videos that will help to explain the structural or molecular formula if you're confused about that, but this is strictly looking at these models. So there are 4 groups that you can categorize them in. We got the carbohydrates, we got the lipids, the proteins, and then the nucleic acids or the nucleotides. So here what you're seeing is some sample models of the carbohydrates. And when you're looking at this, you should kind of be seeing a mass of black, white, and red. And let me first just explain that the different color beads are representative of different elements. So red, for example, is going to be oxygen, black is going to be carbon, and white is hydrogen. Now I want to go through the individual ones and maybe point out some key features that is going to separate these individual molecules. Okay, so I'm going to move these a little bit out of the way. Okay. The first molecule we're going to look at is going to be glucose. Glucose is a monosaccharide, and it is going to be characterized by having 6 carbons, 12 hydrogens, and 6 oxygen. Glucose is arranged in a circular pattern, okay? So there is actually 6, or hexose, there's 6 different atoms that make up that circle, and we could count them. 1, 2, 3, 4, 5, 6. Okay, so there's going to be 5 carbons and 1 oxygen. And then we have various other things attached. Now you recall that I said that there is 6 carbons, and we can look at that as well, and we can count that. 1, 2, 3, 4, 5, and this one is 6. That's sort of attached to one of the carbons that is in the circle. And again, this is a glucose molecule. So what you want to do is count the number of carbons. You know that there's 6. And look at the number of atoms that make up the circle. In this case, it is 6. 5 carbons, 1 oxygen, and that is going to tell you that this is a glucose molecule. Now, a molecule that is an isomer of glucose is going to be fructose. So I'm going to put that one to the side, and we're going to look at this molecule. And what we can see is that there's also 6 carbons, there is going to be 6 oxygens and 12 hydrogens, but they are arranged differently in space. So the first thing you can look at is the ring. And what we can do is count the number of carbons in that ring. So if I start here with oxygen, 1, 2, 3, 4, 5. The other one was 6, and this one's 5. So this is a pentose. And, but yet it still has 6 carbons. 1, 2, 3, 4, 5, 6. So a 5-carbon structure with 6 carbons in total is going to be fructose. Again, another monosaccharide. And of course, those are very important in our nutrition and metabolism. We also are going to be looking at 2 other sugars, monosaccharides, that are very important in our body. And I'm going to pull these over here. And let's start with this one. So how would you, when you take a look at this compared to, say, this, how are you differentiating these? So I'm going to explain that. In this, let me tell you that this is ribose. Ribose is a sugar. Ribose is found in the RNA, okay? And ribose has 5 carbons. And it's also going to have 5 oxygens. So we can count that, 1, 2, 3, 4, 5. And remember, glucose had 6, okay? And we can look at that there is 5 oxygens on here. So this is ribose. Now when we pull this one up, okay, I'm going to take that one down. This is deoxyribose, and deoxyribose has 5 carbons, just like ribose, except for the deoxy means that there is 1 less oxygen. And so here we're looking at the 5 carbon, it's a ring structure, but 1, 2, 3, 4 oxygen. And that's the easiest way to differentiate those versus these other sugars. Okay, put those to the side. And now we're going to look at 2 disaccharides, 2 very important disaccharides. All right, so we have these 2, and they look very, very similar. So again, what we want to do is to be able to differentiate the glucose from the fructose. We already have seen that. So basically, this is maltose. Maltose is a glucose plus a glucose that has undergone a reaction, a dehydration reaction. And so we lose a water when they dehydrate, and they become the 2 monosaccharides, a disaccharide. And so when you look at these individual rings, you can see that there is 6 carbons in here, 1, 2, 3, 4, 5, 6. And you can see that this is a structure that has 6 atoms in the ring. And over here, it's the same way on this one. We got a 6-ring structure, and we have 6 carbons. And so we know that this is a glucose plus a glucose, and that's what makes the maltose. On this guy, what we're looking at is, let's look at this ring structure. This is going to be a glucose as well. 1, 2, 3, 4, 5, 6 as far as the ring. And then, of course, it has the 6 carbons. But over here, this is different. This is going to be a fructose. So how do I know again? I have a 5-ring structure with 6 carbons. And so 1, 2, 3, 4, 5, 5-ring structure, but we have the 6 carbons. So we have these 2 that are sort of on the outside. This fructose and this glucose underwent a dehydration reaction, and therefore what we have is sucrose. Again, it's a disaccharide. Okay, that concludes the carbohydrate portion of this.